Here we'll learn the components of a eukaryotic animal cell. To begin, draw a plasma membrane that has two layers. Use a zoomed-in window to show that each layer comprises a row of phospholipids. Thus it's often called the phospholipid bilayer. As well, show that it comprises proteins, cholesterol, carbohydrates. They associate with the bilayer in a variety of ways. Write that the plasma membrane encloses the cell, providing a protective barrier from its external environment and regulates traffic of materials in and out of the cell. Label the external environment as the extracellular space and the internal environment as the intracellular space, otherwise known as the cytosol. The cytosol is the fluid portion of the cytoplasm that does not include organelles. Write that the cytosol is an aqueous, or water-based solution that bathes the organelles and contains a variety of molecules such as ions, metabolites like fatty acids, carbohydrates, and amino acids, as well as proteins such as enzymes. These molecules make it more gel-like than liquid. Before we add organelles, let's learn about an important network within the cell. Write that the eukaryotic cell contains an endomembrane system, which is a select group of membranous organelles that regulate protein trafficking and metabolism. We'll learn its components now and then list them for our review. First, draw the nuclear envelope as a discontinuous double membrane. Label the gaps in the envelope as nuclear pores. Indicate that the nucleus lies within the nuclear envelope. The nucleus is the largest organelle in eukaryotic cells, approximately 5 micrometers in diameter, and visible under a light microscope. Write that the nucleus stores the cell's genetic material, DNA. It also is the site of DNA replication and RNA synthesis, transcription. We'll see that protein synthesis, translation, occurs in the cytosol. Within the nucleus, draw a representative thin strand of chromatin, the uncondensed form of DNA. A nucleolus. Nuclei contain one or more of these non-membranous organelles. Cells that produce a large amount of proteins have a proportionally large nucleolus and a large number of ribosomes. Draw a strand of rRNA within the nucleolus where it's synthesized. Ribosomal proteins assemble in the nucleolus before maturing in the cytoplasm. Next, draw several free ribosomes suspended in the cytosol. Using a zoomed-in window, show that each ribosome comprises two subunits, one large and one small. Write that a ribosome binds to mRNA to synthesize proteins via translation. The ribosome can be suspended in the cytosol or bound to the endoplasmic reticulum. Now draw an endoplasmic reticulum that's continuous with the nuclear envelope. It comprises membranous sacs and tubules called cisternae. Show that the cisternae enclose a space called the ER lumen. The ER membrane separates these luminal spaces from the cytosol. It's continuous with the nuclear envelope. Next, show a representative portion of the ER as covered in ribosomes. Label it rough ER. Bound ribosomes synthesize and secrete proteins either into the lumen where they fold into their native conformation or into the ER membrane to produce transmembrane proteins. Label smooth ER as the portions that lack ribosomes. Now write that the rough ER is a site of protein synthesis, processing, and secretion. Next, write that the smooth ER maintains the following functions: lipid synthesis, production and secretion of steroid hormones, carbohydrate metabolism, detoxification of substances. As a clinical correlation, drugs and/or alcohol can induce the proliferation of smooth ER in liver cells, which accelerates detoxification. Next, let's illustrate how the ER secretes proteins. Draw a transport vesicle in the cytosol. Show that transport vesicles bud from the ER. They keep secretory proteins separate from proteins synthesized in the cytosol. Next, draw a Golgi apparatus as a series of stacked cisternae. Label the side of the Golgi that faces the nucleus as cis. It receives cargo from the ER. Label the opposite side of the Golgi that faces the plasma membrane as trans. Proteins exit the Golgi from the trans face. So show that the transport vesicle fuses with the Golgi apparatus on the cis side. Write that the Golgi modifies, stores, and secretes proteins and lipids that it receives from the ER, and that it synthesizes its own lipids. Cells active in secretion have an extensive Golgi. Next, draw a lysosome, then an endosome. Write that a lysosome is a vesicle that contains hydrolytic enzymes synthesized in the rough ER and processed in the Golgi. Write that an endosome forms when the cell engulfs nutrients or other particles via endocytosis. Use an arrow to show that endosomes mature into lysosomes and digest their cargo. As a clinical correlation, Tay-Sachs disease is a lysosomal storage disease that presents when lysosomes are missing a lipid digesting enzyme. Lipids accumulate in cells because lysosomes cannot digest them. As a result, they impair brain function. Now, this concludes the endomembrane system. To review, write that the endomembrane system includes the nucleus, endoplasmic reticulum, Golgi apparatus, endosomes, and lysosomes. Now in the cytosol, draw the outer membrane of a rod-shaped mitochondrion. This organelle is considered the cell's powerhouse because it generates all of the energy, ATP, necessary for cell function. Using a zoomed-in window, let's draw a mitochondrion. First its outer membrane, and then show that the mitochondrion's inner membrane folds to form cristae. It has a double membrane just like the nuclear envelope. Label the space within the cristae as matrix. Label the space between the outer and inner membranes as inner membrane space. Write that mitochondria synthesize ATP, the cell's energy currency, that they do so via the citric acid cycle and oxidative phosphorylation, which is the coupling of two reactions, the oxidation of nutrients and the phosphorylation of ADP. Mitochondria have electron transport chains and enzymes embedded in their inner membrane, which facilitates these reactions. Finally, show that free ribosomes lie within the mitochondrial matrix. For review, write that two organelles within the cell have double membranes, the nucleus and mitochondria. Now let's draw the remaining cytosolic features of the cell. Draw a peroxisome as a small membranous organelle. Write that they produce hydrogen peroxide from the detoxification or breakdown of various substances such as alcohol. Any leftover hydrogen peroxide is then broken down into water and oxygen by an enzyme called catalase. Peroxisomes do not bud from the endomembrane system like lysosomes do. Now let's draw the cytoskeleton, the structural support for the cell. Draw microfilaments, then intermediate filaments, and then microtubules. Write that the cytoskeleton anchors organelles and provides the structural framework for the cell. Show that the microtubules nucleate at a region called the centrosome. Indicate that it contains two small structures called centrioles, and that the whole structure functions in cell division. This concludes our diagram. Here we'll learn an overview of the concepts of osmosis and osmolarity. These concepts, in addition to the concept of diffusion that we learned before, are important when we later discuss the movement of molecules across a cell membrane. First, start a table to define some key terms. Denote that osmosis is the diffusion of water across a semi-permeable membrane due to differences in solute concentrations. We'll explain this in detail shortly. Denote that osmolarity, also known as osmotic concentration, is a measure of solute concentration, meaning the amount of osmoles of solute per liter. Begin with osmosis. Draw two U-shaped pipes. Use a porous membrane to separate both sides of the pipes. Indicate that it's a semi-permeable membrane, which means it allows some molecules to pass through it, but not others. Indicate that water is present in the left pipe and that it's at equal levels on both sides. Add some solute molecules to the left side of the pipe. They're too large to fit through the membrane. Now add a greater number of solute molecules to the right side of the pipe.
indicate that the left side of the pipe has a low solute concentration and the right side of the pipe has a high solute concentration. It's this difference in solute concentration across a semi-permeable membrane that causes osmosis to occur. In the pipe on the right, indicate that water has moved from the left side, which now has a lower level of water, to the right side, which now has a higher level. Indicate that because the amount of solute remained the same on each side, and it was only the water that moved, the concentration of solute is now the same on both sides of the membrane. Now indicate that we'll discuss the concept of osmolarity, which is the solute concentration measured as the amount of osmoles of solute per liter. Draw one box for ionic compounds in solution, and another for non-ionic compounds in solution. Indicate that we'll be adding 2 moles of sodium chloride to the left box, using 2 combined shapes to distinguish the 2 ions. Because ionic compounds dissociate within solution, within the box, draw the 2 shapes that represent the sodium ions and the 2 shapes that represent the chloride ions. There should be 4 separate shapes in the box. Now indicate that our solution of sodium chloride has 4 osmoles of solute. Above the right box, indicate that we'll be adding 2 moles of glucose to the solution, using a singular shape to represent the glucose molecule. Because glucose does not dissociate in solution, draw only 2 shapes in the box to represent the 2 moles of glucose added. Finally, indicate that our solution of glucose has 2 osmoles of solute. Thus, osmolarity is similar to the concept of molarity, but with a key difference that 1 mole of a substance can actually have more than 1 osmoles in solution, as we've just seen with the ionic compounds in solution. To demonstrate the semi-permeable nature of a cell's plasma membrane, which allows water to freely flow while restricting the movement of other molecules, let's explore what would happen if you drop a red blood cell into solutions with 3 different solute concentrations. Begin with a solution that's isotonic. Draw a normal red blood cell in the isotonic solution. Show that the solution has the same solute concentration as the interior of the cell. Show that water freely diffuses in and out of the cell at an equilibrium. Now show a hypotonic solution. It has a lower solute concentration than the intracellular environment. Indicate that if we placed our red blood cell in a hypotonic environment, in which the red blood cell has a higher solute concentration than the solution, water would diffuse into the cell and cause it to lyse. Lastly, show a hypertonic solution. It has a greater solute concentration than the intracellular environment. Show that in a hypertonic environment, in which the red blood cell has a lower solute concentration than in the solution, our cell would shrivel up. Show that water would leave the cell. This concludes our diagram. Here we'll learn an overview of the concept of diffusion. Understanding this concept, along with the concept of osmosis and osmolarity, which we discuss elsewhere, is important when discussing movement of molecules across a cell membrane. First, start a table to define some key terms. Denote that diffusion is the tendency of a substance to spread out evenly in the available space from an area of high concentration to an area of low concentration. Denote that a solution has one or more substances, solutes, dissolved in another substance, solvent. To visualize diffusion, imagine a glass of water to which a drop of food coloring is added. Notice how the color spreads out from the point the drop hits the water. This is diffusion of the food coloring molecules. Indicate that we'll start with the diffusion of one solute. Draw 3 boxes and indicate that they're filled with water. In each box, draw a membrane with pores vertically down the middle. In the leftmost box, draw small solute molecules entirely on the left side of the membrane. Show that they move randomly. Some of them make it through the pores and some bounce back. Indicate that the net movement of solute molecules is toward the right side of the box, meaning toward the area of low concentration. In the middle box, draw a couple of solute molecules on the right and the majority on the left. Indicate that random movement continues with some molecules on each side making it through the pores and others bouncing back. Again, indicate that the net movement of the solute is still towards the right. In the final box, draw equal amounts of solute on both sides. Indicate that random movement continues, but note that any time a solute molecule passes through a pore, a molecule from the other side also passes through. Indicate that this situation is called equilibrium. Here there is no net movement of molecules from one side to the other. Now let's explore diffusion when 2 or more solutes are present in a solution. Write that the rate of diffusion of 2 or more solutes is independent of each other. Again, draw 3 boxes filled with water. Draw a membrane with pores vertically down the middle of each box. In the leftmost box, draw a few of one type of solute molecule, solute A, on the left side, and more of another type of solute molecule, solute B, on the right side. Again, indicate the random motion of these molecules, with some able to make it through the pores in both directions, and others bouncing back. Indicate that the net movement of solute A is to the right, and the net movement of solute B is to the left. In the middle box, draw equal numbers of solute A molecules on either side of the membrane, and draw a few solute B molecules on the left side of the membrane. Indicate that random movement continues, though now equal numbers of solute A molecules pass back and forth across the membrane, while more solute B molecules are passing to the left than are passing to the right. Indicate that solute A has reached equilibrium, while the net movement of solute B is still towards the left. In the final box, draw equal amounts of solute A on both sides of the membrane and equal amounts of solute B on both sides of the membrane. Indicate that both solutes have now reached equilibrium. Remember that the rate at which solute A reaches equilibrium is the same no matter if solute B is present or not. This concludes our diagram. Here we'll learn the common functional groups in biochemistry and some of their functional roles. These functional groups define common molecules, compounds, and reactions in biochemistry, including biochemical polymers such as proteins, DNA, and carbohydrates. Start at table. Denote that we'll learn the following four groups: carbon-oxygen groups, carbon-nitrogen groups, carbon-sulfur groups, and esters and amides. Next, let's list the individual molecules that are affiliated with each of these groups. Within the carbon-oxygen group are alcohols, aldehydes, ketones, carboxylic acids, ethers, and acid anhydrides. These functional groups are so ubiquitous that they're responsible for almost all biochemical reactions. Our second category of functional groups is carbon-nitrogen groups, including amino groups, primary amines, and quaternary amines. Amines are most notably involved in primary protein structure, which is Here we'll learn the common functional groups in biochemistry and some of their functional roles. These functional groups define common molecules, compounds, and reactions in biochemistry, including biochemical polymers such as proteins, DNA, and carbohydrates. Start a table. Denote that we'll learn the following four groups: carbon-oxygen groups, carbon-nitrogen groups, carbon-sulfur groups, and esters and amides. Next, let's list the individual molecules that are affiliated with each of these groups. Within the carbon-oxygen group are alcohols, aldehydes, ketones, carboxylic acids, ethers, and acid anhydrides. These functional groups are so ubiquitous that they're responsible for almost all biochemical reactions. Our second category of functional groups is carbon-nitrogen groups, including amino groups, primary amines, and quaternary amines. Here we'll learn the common functional groups in biochemistry and some of their functional roles. These functional groups define common molecules, compounds, and reactions in biochemistry, including biochemical polymers such as proteins, DNA, and carbohydrates. Here we will begin to review some of the organic chemistry principles that are important in our understanding of carbohydrate biochemistry, especially as it relates to how the body metabolizes carbohydrate compounds. Specific topics we'll address are the structures and characteristics of aldose and ketose sugars, and the differences between various types of isomers. First, let's make a table. Indicate that carbohydrates are Here we'll learn the common functional groups in biochemistry and some of their functional roles. These functional groups define common molecules, compounds, and reactions in biochemistry, including biochemical polymers such as proteins, DNA, and carbohydrates. Start a table. Denote that we'll learn the following four. Here we'll continue to review some of the organic chemistry principles that are important in our understanding of carbohydrate biochemistry. We'll specifically address the isomers present in carbohydrate biochemistry. Start a table. Write that there are two types of stereoisomers: enantiomers, which differ at all chiral carbons or stereocenters, making them mirror images of each other. Enantiomers have identical physical properties to one another, except that they rotate plane polarized light in opposite directions, as discussed elsewhere. Diastereomers, which differ at one or more, but not all stereocenters. Diastereomers are called epimers if they differ at only one stereocenter in molecules with multiple stereocenters, and are called anomers if they differ at new stereocenters created during ring closing reactions. However, there are diastereomers that are neither epimers or anomers, and they're simply unspecified. We'll now draw examples of epimers, anomers, and non-epimer, non-anomer diastereomers. To illustrate this concept, we'll use aldohexoses. As their name implies, they are aldose sugars with 6 carbons. First, draw a vertical chain of 6 carbons. Label the carbon atoms 1 through 6 from top to bottom. To carbon 1, add a carbonyl group and a hydrogen atom. To carbons 2 and 3, add a hydroxyl group on the left and a hydrogen atom on the right. For carbons 4 and 5, add a hydrogen atom on the left and a hydroxyl group on the right. Finally, on carbon 6, add 2 hydrogen atoms and a hydroxyl group. Indicate that this molecule is D-mannose, a sugar commonly used in the glycosylation of proteins. Notice that there are 4 carbons, 2 through 5, that are all chiral, shown in pink, thus giving 2 to the power of 4, which equals 16, different stereoisomers. Only a few of these are generally considered relevant in human biochemistry. Now let's draw one of the diastereomers of D-mannose to the right of it. Once again, draw a vertical chain of 6 carbons. Label the carbon atoms 1 through 6 from top to bottom. To carbon 1, add a carbonyl group and a hydrogen atom. To carbons 2, 4, and 5, add a hydrogen atom on the left and a hydroxyl group on the right. For carbon 3, add a hydroxyl group on the left and a hydrogen atom on the right. Finally, on carbon 6, add 2 hydrogen atoms and a hydroxyl group. Indicate that this molecule is D-glucose, the primary sugar used in metabolism to generate ATP. Next, let's indicate how D-mannose and D-glucose are both diastereomers and epimers of one another. First, dash a box around both C2s to highlight that D-mannose and D-glucose are diastereomers of each other, since they are not mirror images, but they differ in the spatial arrangement of atoms at carbon 2. Second, indicate that since they only differ at one stereocenter, carbon 2, they are also epimers of each other. Now let's draw D-galactose, because it's a good example of a molecule that is a diastereomer of both D-mannose and D-glucose, but only an epimer of D-glucose. Next to our drawing of D-mannose and D-glucose, draw a third vertical chain of 6 carbons and label the carbon atoms 1 through 6 from top to bottom. Add the carbonyl group and hydrogen to carbon 1 and the 2 hydrogens and hydroxyl group to carbon 6. To carbons 2 and 5, add a hydrogen atom on the left of the carbon and a hydroxyl group on its right. To carbons 3 and 4, do the opposite. Add the hydroxyl group on the left and the hydrogen atom on the right. Indicate that this molecule is D-galactose, another simple sugar used in metabolism and in the formation of carbohydrates. Notice that D-galactose differs from D-mannose at carbons 2 and 4, making it a diastereomer, but not an epimer. On the other hand, D-galactose only differs from D-glucose at carbon 4, making it a diastereomer and an epimer of D-glucose. Finally, we'll draw examples of anomers. Anomers are specifically diastereomers of ring structures. So for these next drawings, we'll be drawing ring-form aldohexoses. First, let's draw the sugar alpha-D-glucose. Draw a hexagon with a pair of parallel sides going horizontally, and
Here
insert
we'll
an
learn an
oxygen
overview of the
atom at
concept of
apex the two
diffusion.
lines on the top
Understanding
right
this
of the
concept,
hexagon.
along with the concept of
Recall from
osmosis
organic
and
chemistry that
osmolarity,
stick
which we
drawings of
discuss
organic
elsewhere, is
compounds,
important when discussing
carbon atoms
movement
are
of
at the end
molecules
of the lines
across a cell
or at the
membrane.
points where the two lines meet.
First, start a table to define
Let's
some
label
key
the carbon
terms.
atoms in this
Note that
molecule.
diffusion is the tendency of a
Moving
substance to
clockwise
spread
from
out
our
evenly
oxygen
in the
atom,
available space.
Label the carbons 1 through 5. Notice that our sixth carbon atom is missing. Let's add it as an attachment to carbon 5 as follows. Draw a vertical line upwards from carbon 5 and add a carbon with 2 hydrogens and a hydroxyl group attached to it. Now let's add the other hydroxyl groups. To carbons